Biological pest control. Biological control or biocontrol is a method of controlling pests, whether pest animals such as insects and mites, weeds, or pathogens affecting animals or plants by using other organisms. 1. It relies on predation, parasitism, herbivora, or other natural mechanisms, but typically also involves an active human management role. It can be an important component of integrated pest management (IPM) programs. Surface hoverfly larvae below feed on aphids above, making them natural biological control agents. A parasitoid wasp, Cotesia congregata, adult with pupal cocoons on its host. A tobacco hornworm, Manduca sexta, green background, an example of a hymenopteran biological control agent. There are 3 basic strategies for biological control: classical importation, where a natural enemy of a pest is introduced in the hope of achieving control; inductive augmentation, in which a large population of natural enemies are administered for quick pest control; and inoculative conservation, in which measures are taken to maintain natural enemies through regular reestablishment. 2. Natural enemies of insects play an important part in limiting the densities of potential pests. There are 3 basic strategies for biological control: classical importation, where a natural enemy of a pest is introduced in the hope of achieving control; inductive augmentation, in which a large population of natural enemies are administered for quick pest control; and inoculative conservation, in which measures are taken to maintain natural enemies through regular reestablishment. 2. Natural enemies of insects play an important part in limiting the densities of potential pests. Biological control agents such as these include predators, parasitoids, pathogens, and competitors. Biological control agents of plant diseases are most often referred to as antagonists. Biological control agents of weeds include seed predators, herbivores, and plant pathogens. Biological control can have side effects on biodiversity through attacks on non-target species by any of the above mechanisms, especially when a species is introduced without a thorough understanding of the possible consequences. The term, biological control, was first used by Harry Scott Smith at the 1919 meeting of the Pacific Slope branch of the American Association of Economic Entomologists, in Riverside, California. 3. It was brought into more widespread use by the entomologist Paul H. de Bach, 1914-1993, who worked on citrus crop pests throughout his life. 4. 5. However, the practice has previously been used for centuries. The first report of the use of an insect species to control an insect pest comes from Nanfang Kao Mu Zhuang, Nanfang Sao Mu Zhuang plants of the southern regions, circa 304 AD, attributed to Western Jin dynasty botanist Jihan, 263-307, in which it is mentioned that Jiaoji people sell ants in their nests attached to twigs looking like thin cotton envelopes, the reddish-yellow ant being larger than normal. Without such ants, southern citrus fruits will be severely insect-damaged. 6. The ants used are known as huang gan, huang equals yellow, gan equals citrus, ants, Oecophilus smaragdina. The practice was later reported by Ling Biao Luyi, late Tang dynasty or early 5 dynasties, in Jilapian by Zhuang Jishu, southern Song dynasty, in the Book of Tree Planting by Yu Zhenmu, Ming dynasty, in the book Guangdong Xingyu, 17th century, Lingnan by Wu Zhenfang, Qing dynasty, in Nanyu Miscellanies by Li Diaoyuan, and others. 6. Biological control techniques as we know them today started to emerge in the 1870s. During this decade, in the U.S., the Missouri state entomologist C.V. Riley and the Illinois state entomologist W. LeBaron began within state redistribution of parasitoids to control crop pests. The first international shipment of an insect as a biological control agent was made by Charles V. Riley in 1873, shipping to France the predatory mites Pyroglyphus phylloxera to help fight the grapevine phylloxera, Dactylus faravidifolii, that was destroying grapevines in France. The United States Department of Agriculture, USDA, initiated research in classical biological control following the establishment of the Division of Entomology in 1881, with C.V. Riley as chief. The first importation of a parasitoidal wasp into the United States was that of the Braconid Cotesia glomerata in 1883-1884, imported. History. From Europe to control the invasive cabbage white butterfly, Pieris rapi. In 1888-1889 the Vadalia beetle, Novius cardinalis, a lady beetle, was introduced from Australia to California to control the cottony cushion scale, Iceria purchasii. This had become a major problem for the newly developed citrus industry in California, but by the end of 1889, the cottony cushion scale population had already declined. This great success led to further introductions of beneficial insects into the U.S. 7. 8. In 1905 the USDA initiated its first large-scale biological control program, sending entomologists to Europe and Japan to look for natural enemies of the spongy moth, Lymantria disper, and the brown-tail moth, Euproctis chrysaria, invasive pests of trees and shrubs. As a result, 9 parasitoids, solitary wasps, of the spongy moth, 7 of the brown-tail moth, and 2 predators of both moths became established in the U.S. Although the spongy moth was not fully controlled by these natural enemies, the frequency, duration, and severity of its outbreaks were reduced and the program was regarded as successful. This program also led to the development of many concepts, principles, and procedures for the implementation of biological control programs. 7, 8, 9. Cactoblastis cactorum larvae feeding on Opuntia prickly pear cacti. Prickly pear cacti were introduced into Queensland, Australia as ornamental plants, starting in 1788. They quickly spread to cover over 25 million hectares of Australia by 1920, increasing by 1 million hectares per year. Digging, burning, and crushing all proved ineffective. Two control agents were introduced to help control the spread of the plant, the cactus moth Cactoblastus cactorum, and the scale insect Dactylopius. Between 1926 and 1931, tens of millions of cactus moth eggs were distributed around Queensland with great success, and by 1932, most areas of prickly pear had been destroyed. 10. The first reported case of a classical biological control attempt in Canada involves the parasitoidal wasp Trichogramma minutum. Individuals were caught in New York State and released in Ontario Gardens in 1882 by William Saunders, a trained chemist and first director of the Dominion Experimental Farms, for controlling the invasive Corontorm nematis ribacea. Between 1884 and 1908, the first Dominion entomologist, James Fletcher, continued introductions of other parasitoids and pathogens for the control of pests in Canada. 11. Types of biological pest control. There are three basic biological pest control strategies: importation, classical biological control, augmentation, and conservation. 12. Importation. Rhodelia cardinalis, the Batelia beetle, was imported from Australia to California in the 19th century, successfully controlling cottony cushion scale on orange trees. Importation or classical biological control involves the introduction of a pest's natural enemies to a new locale where they do not occur naturally. Early instances were often unofficial and not based on research, and some introduced species became serious pests themselves. 13. To be most effective at controlling a pest, a biological control agent requires a colonizing ability which allows it to keep pace with changes to the habitat in space and time. Control is greatest if the agent has temporal persistence so that it can maintain its population even in the temporary absence of the target species, and if it is an opportunistic forager, enabling it to rapidly exploit a pest population. 14. One of the earliest successes was in controlling Iceria purchasii, cottony cushion scale, in Australia, using a predatory insect Rhodelia cardinalis, the Batelia beetle. This success was repeated in California using the beetle and a parasitoidal fly, Cryptochaetum iceriae. 15 other successful cases include the control of Antonina graminis in Texas by Neodusmetia sengwani in the 1960s. 16. Damage from Hyperapostica, the alfalfa weevil, a serious introduced pest of forage, was substantially reduced by the introduction of natural enemies. 20 years after their introduction, the population of weevils in the alfalfa area treated for alfalfa weevil in the northeastern United States remained 75% down. 17. The invasive species Alternanthera phylloxeroides, alligator weed, was controlled in Florida, U.S., by introducing alligator weed flea beetle. Alligator weed was introduced to the United States from South America. It takes root in shallow water, interfering with navigation, irrigation, and flood control. The alligator weed flea beetle and two other biological controls were released in Florida, greatly reducing the amount of land covered by the plant. 18. Another aquatic weed, the giant Salvinia, Salvinia molesta, is a serious pest, covering waterways, reducing water flow and harming native species. Control with the Salvinia weevil, Certobagus salvinii, and the Salvinia stem borer moth, Samea multiplicalis, is effective in warm climates. 1920, and in Zimbabwe, a 99% control of the weed was obtained over a 2-year period. 21. Small, commercially reared parasitoidal wasps, 12 Trichogramma austrinii, provide limited and erratic control of the European corn borer, Austrinia nubilalis, a serious pest. Careful formulations of the bacterium Bacillus thuringiensis are more effective. The O. nubilalis integrated control releasing Trichogramma brassicae, egg parasitoid, and later Bacillus thuringiensis subs. Kerstaki, larvicide effect, reduce pest damages more than insecticide treatments. 22. The population of Levuana iridescens, the Levuana moth, a serious coconut pest in Fiji, was brought under control by a classical biological control program in the 1920s. 23. Augmentation. Hippotomia convergens, the convergent lady beetle, is commonly sold for biological control of aphids. Augmentation involves the supplemental release of natural enemies that occur in a particular area, boosting the naturally occurring populations there. In inoculative release, small numbers of the control agents are released at intervals to allow them to reproduce, in the hope of setting up longer-term control and thus keeping the pest down to a low level, constituting prevention rather than cure. In inundative release, in contrast, large numbers are released in the hope of rapidly reducing a damaging pest population, correcting a problem that has already arisen. Augmentation can be effective, but is not guaranteed to work, and depends on the precise details of the interactions between each pest and control agent. 24. An example of inoculative release occurs in the horticultural production of several crops in greenhouses. Periodic releases of the parasitoidal wasp, Encarsia formosa, are used to control greenhouse whitefly. 25. While the predatory mite Phytoceolus persimilis is used for control of the two-spotted spider mite. 26. The egg parasite Trichogramma is frequently released inundatively to control harmful moths. New way for inundative releases are now introduced, i.e. use of drones. Egg parasitoids are able to find the eggs of the target host by means of several cues. Chiromones were found on moth scales. Similarly, Bacillus thuringiensis and other microbial insecticides are used in large enough quantities for a rapid effect. 24. Recommended release rates for Trichogramma in vegetable or field crops range from 5,000 to 200,000 per acre, 1 to 50 per square meter per week according to the level of pest infestation. 27. Similarly, nematodes that kill insects that are entomopathogenic are released at rates of millions and even billions per acre for control of certain soil-dwelling insect pests. 28. Conservation. The conservation of existing natural enemies in an environment is the third method of biological pest control. 29. Natural enemies are already adapted to the habitat and to the target pest, and their conservation can be simple and cost-effective, as when nectar-producing crop plants are grown in the borders of rice fields. These provide nectar to support parasitoids and predators of planthopper pests and have been demonstrated to be so effective, reducing pest densities by 10 or even 100-fold, that farmers sprayed 70% less insecticides and enjoyed yields boosted by 5%. 30. Predators of aphids were similarly found to be present in tussock grasses by field boundary hedges in England, but they spread too slowly to reach the centers of fields. Control was improved by planting a meter-wide strip of tussock grasses in field centers, enabling aphid predators to overwinter there. 29. An inverted flower pot filled with straw to attract earwigs. Cropping systems can be modified to favor natural enemies, a practice sometimes referred to as habitat manipulation. Providing a suitable habitat, such as a shelter belt, hedgerow, or beetle bank where beneficial insects such as parasitoidal wasps can live and reproduce, can help ensure the survival of populations of natural enemies. Things as simple as leaving a layer of fallen leaves or mulch in place provides a suitable food source for worms and provides a shelter for insects, in turn being a food source for such beneficial mammals as hedgehogs and shrews. Compost piles and stacks of wood can provide shelter for invertebrates and small mammals. Long grass and ponds support amphibians. Not removing dead annuals and non-hardy plants in the autumn allow insects to make use of their hollow stems during winter. 31. In California, prune trees are sometimes planted in grape vineyards to provide an improved overwintering habitat or refuge for a key grape pest parasitoid. 32. The providing of artificial shelters in the form of wooden caskets, boxes or flower pots is also sometimes undertaken, particularly in gardens, to make a cropped area more attractive to natural enemies. For example, earwigs are natural predators that can be encouraged in gardens by hanging upside-down flower pots filled with straw or wood wool. Green lacewings can be encouraged by using plastic bottles with an open bottom and a roll of cardboard inside. Birdhouses enable insectivorous birds to nest. The most useful birds can be attracted by choosing an opening just large enough for the desired species. 31. In cotton production, the replacement of broad-spectrum insecticides with selective control measures such as Bt cotton can create a more favorable environment for natural enemies of cotton pests due to reduced insecticide exposure risk. Such predators or parasitoids can control pests not affected by the Bt protein. Reduced prey quality and abundance associated with increased control from Bt cotton can also indirectly decrease natural enemy populations in some cases, but the percentage of pests eaten or parasitized in Bt and non-Bt cotton are often similar. 33. Biological control agents. Predators. Predatory lacewings are available from biocontrol dealers. Predators are mainly free-living species that directly consume a large number of prey during their whole lifetime. Given that many major crop pests are insects, many of the predators used in biological control are insectivorous species. Lady beetles, and in particular their larvae which are active between May and July in the northern hemisphere, are voracious predators of aphids, and also consume mites, scale insects and small caterpillars. The spotted lady beetle Caleongilla maculata is also able to feed on the eggs and larvae of the Colorado potato beetle Leptinotarsa disseminata. 34. The larvae of many hoverfly species principally feed upon aphids, one larva devouring up to 400 in its lifetime. Their effectiveness in commercial crops has not been studied. 35. The running crab spider Phylodromus cespidum also prey heavily on aphids, and act as a biological control agent in European fruit orchards. 36. Predatory Polistes wasp searching for bollworms or other caterpillars on a cotton plant. Several species of entomopathogenic nematode are important predators of insect and other invertebrate pests. 37. 38. Entomopathogenic nematodes form a stress-resistant stage known as the infective juvenile. These spread in the soil and infect suitable insect hosts. Upon entering the insect they move to the hemolymph where they recover from their stagnated state of development and release their bacterial symbionts. The bacterial symbionts reproduce and release toxins, which then kill the host insect. 38. 39. Phasmarhabditis hermaphrodita is a microscopic nematode that kills slugs. Its complex life cycle includes a free-living, infective stage in the soil where it becomes associated with a pathogenic bacteria such as Moraxella osloensis. The nematode enters the slug through the posterior mantle region, thereafter feeding and reproducing inside, but it is the bacteria that kill the slug. The nematode is available commercially in Europe and is applied by watering onto moist soil. 40. Entomopathogenic nematodes have a limited shelf life because of their limited resistance to high temperature and dry conditions. 39. The type of soil they are applied to may also limit their effectiveness. 38. Species used to control spider mites include the predatory mites Phytoceolus persimilis, 41, Neoscilus californicus, 42, and Amblysius cucumaris, the predatory midge Feltiella acarasuga, 42, and a ladybird Stethorus punctillum. 42. The bug Aureus insidiosus has been successfully used against the two-spotted spider mite and the western flower thrips, Franklinella occidentalis. 43. Predators including Cactoblastis cactorum, mentioned above, can also be used to destroy invasive plant species. As another example, the poison hemlock moth, Agonopteryx alstromeriana, can be used to control poison hemlock, Conium maculatum. During its larval stage, the moth strictly consumes its host plant, poison hemlock, and can exist at hundreds of larvae per individual host plant, destroying large swaths of the hemlock. 44. The parasitoid wasp Aleodes indiscretus parasitizing a spongy moth caterpillar, a serious pest of forestry. 45. For rodent pests, cats are effective biological control when used in conjunction with reduction of harborage, hiding locations. 46, 47, 48. While cats are effective at preventing rodent population explosions, they are not effective for eliminating pre-existing severe infestations. 48. Barn owls are also sometimes used as biological rodent control. 49. Although there are no quantitative studies of the effectiveness of barn owls for this purpose, 50. They are known rodent predators that can be used in addition to or instead of cats. 51. 52. They can be encouraged into an area with nest boxes. 53. 54. In Honduras, where the mosquito Aedes aegypti was transmitting dengue fever and other infectious diseases, biological control was attempted by a community action plan. Copepods, baby turtles, and juvenile tilapia were added to the wells and tanks where the mosquito breeds and the mosquito larvae were eliminated. 55. Even amongst arthropods usually thought of as obligate predators of animals, especially other arthropods, floral food sources, nectar and to a lesser degree pollen, are often useful adjunct sources. 56. It had been noticed in one study, 57, that adult Adalia bipunctata predator and common biocontrol of Fs. jacuniella could survive on flowers but never completed its life cycle, so a meta-analysis 56 was done to find such an overall trend in previously published data, if it existed. In some cases floral resources are outright necessary. 56. Overall, floral resources, and an imitation, i.e. sugar water, increase longevity and fecundity, meaning even predatory population numbers can depend on non-prey food abundance. 56. Thus biocontrol population maintenance and success may depend on nearby flowers. 56. Parasitoids. Parasitoids lay their eggs on or in the body of an insect host, which is then used as a food for developing larvae. The host is ultimately killed. Most insect parasitoids are wasps or flies, and many have a very narrow host range. The most important groups are the ichneummonid wasps, which mainly use caterpillars as hosts, braconid wasps, which attack caterpillars and a wide range of other insects including aphids, chalcidoid wasps, which parasitize eggs and larvae of many insect species, and tachnid flies, which parasitize a wide range of insects including caterpillars, beetle adults and larvae, and true bugs. 58. Parasitoids are most effective at reducing pest populations when their host organisms have limited refuges to hide from them. 59. Encarsia formosa, widely used in greenhouse horticulture, was one of the first biological control agents developed. Life cycles of greenhouse whitefly and its parasitoid wasp Encarsia formosa. Parasitoids. Parasitoids are among the most widely used biological control agents. Commercially, there are two types of rearing systems: short-term daily output with high production of parasitoids per day, and long-term, low daily output systems. 60. In most instances, production will need to be matched with the appropriate release dates when susceptible host species at a suitable phase of development will be available. 61. Larger production facilities produce on a year-long basis, whereas some facilities produce only seasonally. Rearing facilities are usually a significant distance from where the agents are to be used in the field, and transporting the parasitoids from the point of production to the point of use can pose problems. 62. Shipping conditions can be too hot, and even vibrations from planes or trucks can adversely affect parasitoids. 60. Encarsia formosa is a small parasitoid wasp attacking whiteflies, sap-feeding insects which can cause wilting and black sooty molds in glasshouse vegetable and ornamental crops. It is most effective when dealing with low-level infestations, giving protection over a long period of time. The wasp lays its eggs in young whitefly scales, turning them black as the parasite larva pupate. 25. Gonatoceras ashmeti, Hymenoptera. Mimeridae has been introduced to control the glassy-winged sharpshooter Homolodisca vitripennis, Hemiptera, Cicadellidae in French Polynesia and has successfully controlled approximately 95% of the pest density. 63. The eastern spruce budworm is an example of a destructive insect in fir and spruce forests. Birds are a natural form of biological control, but the Trichogramma minudum, a species of parasitic wasp, has been investigated as an alternative to more controversial chemical controls. 64. There are a number of recent studies pursuing sustainable methods for controlling urban cockroaches using parasitic wasps. 65. 66. Since most cockroaches remain in the sewer system and sheltered areas which are inaccessible to insecticides, employing active hunter wasps is a strategy to try and reduce their populations. Pathogens. Pathogenic microorganisms include bacteria, fungi, and viruses. They kill or debilitate their host and are relatively host-specific. Various microbial insect diseases occur naturally, but may also be used as biological pesticides. 67. When naturally occurring, these outbreaks are density-dependent in that they generally only occur as insect populations become denser. 68. The use of pathogens against aquatic weeds was unknown until a groundbreaking 1972 proposal by Zettler and Freeman. Up to that point, biocontrol of any kind had not been used against any water weeds. In their review of the possibilities, they noted the lack of interest and information thus far, and listed what was known of pests, whether pathogens or not. They proposed that this should be relatively straightforward to apply in the same way as other biocontrols. 69. And indeed in the decades since, the same biocontrol methods that are routine on land have become common in the water. Bacteria. Bacteria used for biological control infect insects via their digestive tracts, so they offer only limited options for controlling insects with sucking mouth parts such as aphids and scale insects. 70. Bacillus thuringiensis, a soil-dwelling bacterium, is the most widely applied species of bacteria used for biological control, with at least 4 subspecies used against lepidopteran moth, butterfly coleopteran beetle, and dipteran true fly insect pests. The bacterium is available to organic farmers in sachets of dried spores which are mixed with water and sprayed onto vulnerable plants such as brassicas and fruit trees. 71. 72. Genes from B. thuringiensis have also been incorporated into transgenic crops, making the plants express some of the bacterium's toxins, which are proteins. These confer resistance to insect pests and thus reduce the necessity for pesticide use. 73. If pests develop resistance to the toxins in these crops, B. thuringiensis will become useless in organic farming also. 74. 72. The bacterium Pinobacillus populi, which causes milky spore disease, has been found useful in the control of Japanese beetle, killing the larva. It is very specific to its host species and is harmless to vertebrates and other invertebrates. 75. Bacillus spp. M1 fluorescent pseudomonids, M1, and streptomycetes are controls of various fungal pathogens. M2. Columbia mosquito control. The largest ever deployment of Wolbachia-infected aegypti mosquitoes reduced dengue incidence by 94–97% in the Colombian cities of Bello, Medellín, and Itagüí. The project was executed by nonprofit World Mosquito Program, WMP. Wolbachia prevents mosquitoes from transmitting viruses such as dengue and Zika. The insects pass the bacteria onto their offspring. The project covered a combined area of 135 square kilometers, 52 square miles, home to 3.3 million people. Most of the project area reached the target of infecting 60% of local mosquitoes. The technique is not endorsed by WHO. 76. Fungi. Green peach aphid, a pest in its own right and a vector of plant viruses, killed by the fungus Pandora neoaphidus zygomycota. Entomophthorals, scale bar equals 0.3 millimeters. Entomopathogenic fungi, which cause disease in insects, include at least 14 species that attack aphids. 77. Beauveria bassiana is mass-produced and used to manage a wide variety of insect pests including whiteflies, thrips, aphids and weevils. 78. Lecanicilium spp. Are deployed against whiteflies, thrips and aphids. Metarisum spp. Are used against pests including beetles, locusts and other grasshoppers, hemiptera, and spider mites. Poecilomyces fumosroseus is effective against whiteflies, thrips and aphids. Purpureocilium lilicinus is used against root knot nematodes, and 89 Trichoderma species against certain plant pathogens. M3 Trichoderma viride has been used against Dutch elm disease, and has shown some effect in suppressing silverleaf, a disease of stone fruits caused by the pathogenic fungus Chondrostrium purpureum. 79. Pathogenic fungi may be controlled by other fungi, or bacteria or yeasts, such as Gliocladium spp, Mycoparasitic Pythium spp, binucleotypes of Rhizoctonia spp, and Ledosauria spp. The fungi Cordyceps and Metacordyceps are deployed against a wide spectrum of arthropods. 80. Entomophaga is effective against pests such as the green peach aphid. 81. Several members of Chytridiomycota and Blastocladiomycota have been explored as agents of biological control. 82. 83. From Chytridiomycota, Synchytrium solstitial is being considered as a control agent of the yellow star thistle, Centaurea solstitialis, in the United States. 84. Viruses. Baculoviruses are specific to individual insect host species and have been shown to be useful in biological pest control. For example, the Lymantria dispar multicapsid nuclear polyhedrosis virus has been used to spray large areas of forest in North America where larvae of the spongy moth are causing serious defoliation. The moth larvae are killed by the virus they have eaten and die, the disintegrating cadavers leaving virus particles on the foliage to infect other larvae. 85. A mammalian virus. The rabbit hemorrhagic disease virus was introduced to Australia to attempt to control the European rabbit populations there. 86. It escaped from quarantine and spread across the country, killing large numbers of rabbits. Very young animals survived, passing immunity to their offspring in due course and eventually producing a virus-resistant population. 87. Introduction into New Zealand in the 1990s was similarly successful at first, but a decade later, immunity had developed and populations had returned to pre-RHD levels. 88. RNA mycoviruses are controls of various fungal pathogens. M2. Mycota. Legenidium giganteum is a waterborne mold that parasitizes the larval stage of mosquitoes. When applied to water, the motile spores avoid unsuitable host species and search out suitable mosquito larval hosts. This mold has the advantages of a dormant phase, resistant to desiccation, with slow release characteristics over several years. Unfortunately, it is susceptible to many chemicals used in mosquito abatement programs. 89. Competitors. The legume vine Mucuna periens is used in the countries of Benin and Vietnam as a biological control for problematic Imperata cylindrica grass. The vine is extremely vigorous and suppresses neighboring plants by outcompeting them for space and light. Mucuna periens is said not to be invasive outside its cultivated area. 90. Desmodium uncinnatum can be used in push-pull farming to stop the parasitic plant, witchweed, striga. 91. The Australian bushfly, Musca vetustissima, is a major nuisance pest in Australia, but native decomposers found in Australia are not adapted to feeding on cow dung, which is where bushflies breed. Therefore, the Australian Dung Beetle Project 1965-1985, led by George Bornemisha of the Commonwealth Scientific and Industrial Research Organisation, released 49 species of dung beetle, to reduce the amount of dung and therefore also the potential breeding sites of the fly. 92. Combined use of parasitoids and pathogens. In cases of massive and severe infection of invasive pests, techniques of pest control are often used in combination. An example is the emerald ash borer, Agrillus planopennis, an invasive beetle from China, which has destroyed tens of millions of ash trees in its introduced range in North America. As part of the campaign against it, from 2003 American scientists and the Chinese Academy of Forestry searched for its natural enemies in the wild, leading to the discovery of several parasitoid wasps, namely Tetrastachys planopennici, a gregarious larval endoparasitoid, Ubius agrilli, a solitary, parthenogenic egg parasitoid, and Spathius agrilli, a gregarious larval ectoparasitoid. These have been introduced and released into the United States of America as a possible biological control of the emerald ash borer. Initial results for Tetrastachys planopennici have shown promise, and it is now being released along with Bovaria bassiana, a fungal pathogen with known insecticidal properties. 93, 94, 95. Secondary plants. In addition, biological pest control sometimes makes use of plant defenses to reduce crop damage by herbivores. Techniques include polyculture, the planting together of two or more species such as a primary crop and a secondary plant, which may also be a crop. This can allow the secondary plant's defensive chemicals to protect the crop planted with it. 96. Target pests. Fungal pests. Botrytis cinerea on lettuce, by Fusarium spp. And Penicillium claviform, on grape and strawberry by Trichoderma spp. On strawberry by Cladosporium herbarum, on Chinese cabbage by Bacillus brevis, and on various other crops by various yeasts and bacteria. Sclerotinia sclerotiorum by several fungal biocontrols. Fungal pod infection of snap bean by Trichoderma hamadum if before or concurrent with infection. M4 Cryphonectria parasitica, Geumonomyces graminis, Sclerotinia spp. And Ophiostoma novo ulmi by viruses. M2 various powdery mildews and rusts by various Bacillus spp. And fluorescent pseudomonids. M1 Cultitrichum orbiculaire will suppress further infection by itself if manipulated to produce plant-induced systemic resistance by infected the lowest leaf. M5. Difficulties. Many of the most important pests are exotic, invasive species that severely impact agriculture, horticulture, forestry, and urban environments. They tend to arrive without their co-evolved parasites, pathogens and predators, and by escaping from these, populations may soar. Importing the natural enemies of these pests may seem a logical move, but this may have unintended consequences. Regulations may be ineffective and there may be unanticipated effects on biodiversity, and the adoption of the techniques may prove challenging because of a lack of knowledge among farmers and growers. 97. Side effects. Biological control can affect biodiversity 14 through predation. Biological control can affect biodiversity 14 through predation, parasitism, pathogenicity, competition, or other attacks on non-target species. 98. An introduced control does not always can affect biodiversity 14 through predation, parasitism, pathogenicity, competition, or other attacks on non-target species. 98. An introduced control does not always target only the intended pest species, it can also target native species. 99. In Hawaii during the 1940s parasitic wasps were introduced to control a lepidopteran pest and the wasps are still found there today. This may have a negative impact on the native ecosystem, however, host range and impacts need to be studied before declaring their impact on the environment. 100. Cane toad introduced into Australia 1935, spread from 1940 to 1980. It was ineffective as a control agent. Its distribution has continued to widen since 1980. Vertebrate animals tend to be generalist feeders, and seldom make good biological control agents. Many of the classic cases of biocontrol gone awry involve vertebrates. For example, the cane toad, Rhinella marina, was intentionally introduced to Australia to control the greyback cane beetle, Dermalepida albohirtum, 101, and other pests of sugar cane. 102 toads were obtained from Hawaii and bred in captivity to increase their numbers until they were released into the sugar cane fields of the tropic north in 1935. It was later discovered that the toads could not jump very high and so were unable to eat the cane beetles which stayed on the upper stalks of the cane plants. However, the toad thrived by feeding on other insects and soon spread very rapidly. It took over native amphibian habitat and brought foreign disease to native toads and frogs, dramatically reducing their populations. Also, when it is threatened or handled, the cane toad releases poison from parotid glands on its shoulders. Native Australian species such as goannas, tiger snakes, dingoes and northern quolls that attempted to eat the toad were harmed or killed. However, there has been some recent evidence that native predators are adapting, both physiologically and through changing their behavior, so in the long run, their populations may recover. 102. Rhinocilus conicus, a seed-feeding weevil, was introduced to North America to control exotic musk thistle, Cardus nudens, and Canadian thistle, Cirsium arvens. However, the weevil also attacks native thistles, harming such species as the endemic plat thistle, Cirsium neomexicanum, by selecting larger plants, which reduce the gene pool, reducing seed production and ultimately threatening the species' survival. 103. Similarly, the weevil Larinus planus was also used to try to control the Canadian thistle, but it damaged other thistles as well. 104. 105. This included one species classified as threatened. 106. The small Asian mongoose, Herpestus javanicus, was introduced to Hawaii in order to control the rat population. However, the mongoose was diurnal, and the rats emerged at night. The mongoose, therefore, preyed on the endemic birds of Hawaii, especially their eggs, more often than it ate the rats, and now both rats and mongooses threaten the birds. This introduction was undertaken without understanding the consequences of such an action. No regulations existed at the time, and more careful evaluation should prevent such releases now. 107. The sturdy and prolific eastern mosquitofish Gambusia holbrooki is a native of the southeastern United States and was introduced around the world in the 1930s and 40s to feed on mosquito larvae and thus combat malaria. However, it has thrived at the expense of local species, causing a decline of endemic fish and frogs through competition for food resources, as well as through eating their eggs and larvae. 108. In Australia, control of the mosquitofish is the subject of discussion. In 1989, researchers A. H. Ardington and L. L. Lloyd stated that biological population control is well beyond present capabilities. 109. Grower education. A potential obstacle to the adoption of biological pest control measures is that growers may prefer to stay with the familiar use of pesticides. However, pesticides have undesired effects, including the development of resistance among pests, and the destruction of natural enemies. These may in turn enable outbreaks of pests of other species than the ones originally targeted, and on crops at a distance from those treated with pesticides. 110. One method of increasing grower adoption of biocontrol methods involves letting them learn by doing, for example showing them simple field experiments, enabling them to observe the live predation of pests, or demonstrations of parasitized pests. In the Philippines, early season sprays against leaf folder caterpillars were common practice, but growers were asked to follow a rule of thumb of not spraying against leaf folders for the first 30 days after transplanting. Participation in this resulted in a reduction of insecticide use by 1/3 and a change in grower perception of insecticide use. 111. Related techniques. Related to biological pest control is the technique of introducing sterile individuals into the native population of some organism. This technique is widely practiced with insects. Related to biological pest control is the technique of introducing sterile individuals into the native population of some organism. This technique is widely practiced with insects. A large number of males sterilized by radiation are released into the environment, which proceed to compete with the native males for females. Those females that copulate with the sterile males will lay infertile eggs, resulting in a decrease in the size of the population. Over time, with repeated introductions of sterile males, this could result in a significant decrease in the size of the organism's population. Hymenoptera is a large order of insects, comprising the sawflies, wasps, bees, and ants. Over 150,000 living species of Hymenoptera have been described, 3 in addition to over 2,000 extinct ones. 4 Many of the species are parasitic. Females typically have a special ovipositor for inserting eggs into hosts or places that are otherwise inaccessible. This ovipositor is often modified into a stinger. The young develop through holometabolism, complete metamorphosis, that is, they have a worm-like larval stage and an inactive pupal stage before they reach adulthood. The name Hymenoptera refers to the wings of the insects, but the original derivation is ambiguous. 542 All references agree that the derivation involves the ancient Greek pteron for wing. 6 The ancient Greek hymen for membrane provides a plausible etymology for the term because species in this order have membranous wings. 6 However, a key characteristic of this order is that the hindwings are connected to the forewings by a series of hooks. Thus, another plausible etymology involves Hymen, the ancient Greek god of marriage, as these insects have married wings in flight. Another suggestion for the inclusion of Hymen is the myth of Melissa, a nymph with a prominent role at the wedding of Zeus. Molecular analysis finds that Hymenoptera is the earliest branching group of Holometabola. 7. Hymenoptera. Temporal range, Triassic present. Hymenopterans from different families, clockwise from top left. Red imported fire ant. Formicidae, Vespula vulgaris, Vespidae, Tenthrodopsis sordida, Tenthredinidae, and western honeybee, Apidae. Scientific classification domain. Eukaryota kingdom. Animalia phylum. Arthropoda class. Insecta unranked, Holometabola superorder. Hymenopterida order. Hymenoptera. Linnaeus, 1758. Suborders. Symphyta, sawflies, Paraphyletic, Apocrita, wasps, bees and ants. Etymology. Evolution. Holometabola. Hymenoptera. Neuroptera, lacewings. Hymenoptera originated in the Triassic, with the oldest fossils belonging to the family Xylidae. Social hymenopterans appeared during the Cretaceous. 8. The evolution of this group has been intensively studied by Alex Rasmussen, Michael S. Engel, and others. 9. Phylogenetic relationships within the Hymenoptera, based on both morphology and molecular data, have been intensively studied since 2000. 10. In 2023, a molecular study 10 based on the analysis of ultra-conserved elements confirmed many previous findings and produced a relatively robust phylogeny of the whole order. Basal superfamilies are shown in the cladogram below. Hymenoptera. Hymenoptera. Tenthredinoidea. Xyloidea, Triassic present. Pamphyloidea. Onychocaridia. Siracoidea, horntails or wood wasps. Ziphydrioidea, wood wasps. Cephoidea, stem sawflies. Parasitism. Orosoidea, parasitic wood wasps. Wasp waste, Apocrita, ants, bees, wasps. 200 Maya. 250 Maya. Symphyta, red bar, are paraphyletic as Apocrita are excluded. Bombus muscorum drinking nectar with its long proboscis. Hymenopterans range in size from very small to large insects, and usually have 2 pairs of wings. Hymenopterans range in size from very small to large insects, and usually have 2 pairs of wings. Their mouth parts are adapted for chewing, with well-developed mandibles, ectognathous mouthparts. Many species have further developed the mouthparts into a lengthy proboscis, with which they can drink liquids, such as nectar. They have large compound eyes, and typically 3 simple eyes, ocelli. The forward margin of the hind wing bears a number of hooked bristles, or hamuli, which lock onto the forewing, keeping them held together. The smaller species may have only 2 or 3 hamuli on each side, but the largest wasps may have a considerable number, keeping the wings gripped together especially tightly. Hymenopteran wings have relatively few veins compared with many other insects, especially in the smaller species. In the more ancestral hymenopterans, the ovipositor is blade-like, and has evolved for slicing plant tissues. In the majority, however, it is modified for piercing, and, in some cases, is several times the length of the body. In some species, the ovipositor Symphyta. Anatomy has become modified as a stinger, and the eggs are laid from the base of the structure, rather than from the tip, which is used only to inject venom. The sting is typically used to immobilize prey, but in some wasps and bees may be used in defense. 8. Hymenopteran larvae typically have a distinct head region, 3 thoracic segments, and usually 9 or 10 abdominal segments. In the suborder Symphyta, the eruciform larvae resemble caterpillars in appearance, and like them, typically feed on leaves. They have large chewing mandibles, 3 pairs of thoracic limbs, and, in most cases, 6 or 8 abdominal prolegs. Unlike caterpillars, however, the prolegs have no grasping spines, and the antennae are reduced to mere stubs. Symphytan larvae that are wood borers or stem borers have no abdominal legs, and the thoracic legs are smaller than those of non-borers. With rare exceptions, larvae of the suborder Apocrita have no legs and are maggot-like in form, and are adapted to life in a protected environment. This may be the body of a host organism, or a cell in a nest, where the adults will care for the larva. In parasitic forms, the head is often greatly reduced and partially withdrawn into the prothorax, anterior part of the thorax. Sense organs appear to be poorly developed, with no ocelli, very small or absent antennae, and tooth-like, sickle-like, or spine-like mandibles. They are also unable to defecate until they reach adulthood due to having an incomplete digestive tract, a blind sac presumably to avoid contaminating their environment. With rare exceptions, larvae of the suborder Apocrita have no legs and are maggot-like in form, and are adapted to life in a protected environment. This may be the body of a host organism, or a cell in a nest, where the adults will care for the larva. In parasitic forms, the head is often greatly reduced and partially withdrawn into the prothorax, anterior part of the thorax. Sense organs appear to be poorly developed, with no ocelli, very small or absent antennae, and tooth-like, sickle-like, or spine-like mandibles. They are also unable to defecate until they reach adulthood due to having an incomplete digestive tract, a blind sac presumably to avoid contaminating their environment. 8. The larvae of stinging forms, Aculida, generally have 10 pairs of spiracles, or breathing pores, whereas parasitic forms usually have 9 pairs present. 11. Reproduction. Sex determination. Among most or all hymenopterans, sex is determined by the number of chromosomes an individual possesses. 12 fertilized eggs get 2 sets of chromosomes, one from each parent's respective gametes, and develop into diploid females, while unfertilized eggs only contain 1 set, from the mother, and develop into haploid males. The act of fertilization is under the voluntary control of the egg-laying female, giving her control of the sex of her offspring. 8. This phenomenon is called haplodiploidy. However, the actual genetic mechanisms of haplodiploid sex determination may be more complex than simple chromosome number. In many Hymenoptera, sex is determined by a single gene locus with many alleles. 12. In these species, haploids are male and diploids heterozygous at the sex locus or female, but occasionally a diploid will be homozygous at the sex locus and develop as a male, instead. This is especially likely to occur in an individual whose parents were siblings or other close relatives. Diploid males are known to be produced by inbreeding in many ant, bee, and wasp species. Diploid biparental males are usually sterile, but a few species that have fertile diploid males are known. 13. One consequence of haplodiploidy is that females on average have more genes in common with their sisters than they do with their daughters. Because of this, cooperation among kindred females may be unusually advantageous and has been hypothesized to contribute to the multiple origins of eusociality within this order. 8.14 In many colonies of bees, ants, and wasps, worker females will remove eggs laid by other workers due to increased relatedness to direct siblings, a phenomenon known as worker policing. 15. Another consequence is that hymenopterans may be more resistant to the deleterious effects of inbreeding. As males are haploid, any recessive genes will automatically be expressed, exposing them to natural selection. Thus, the genetic load of deleterious genes is purged relatively quickly. 16. The litoki. Some hymenopterans take advantage of parthenogenesis, the creation of embryos without fertilization. The litoki is a particular form of parthenogenesis in which female embryos are created without fertilization. The form of the litoki in hymenopterans is a kind of automixis in which two haploid products, proto-eggs, from the same meiosis fuse to form a diploid zygote. This process tends to maintain heterozygosity in the passage of the genome from mother to daughter. It is found in several ant species including the desert ant Cataglyphus cursor, 17 the clonal raider ant Sarapachys baroi, 18 the predaceous ant Platythyria punctata, 19 and the electric ant, little fire ant Wasmannia oropunctata. 20 it also occurs in the Cape honeybee Apis mellifera capensis. 21. Oocytes that undergo automixis with central fusion often have a reduced rate of crossover recombination, which helps to maintain heterozygosity and avoid inbreeding depression. Species that display central fusion with reduced recombination include the ants Platythyria punctata, 19, and Wasmannia oropunctata, 20, and the Cape honeybee Apis mellifera capensis. 21. In A.M. capensis, the recombination rate during meiosis is reduced more than tenfold. 21. In W. oropunctata the reduction is 45-fold. 20. Single queen colonies of the narrow-headed ant Formica execta illustrate the possible deleterious effects of increased homozygosity. Colonies of this species which have more homozygous queens will age more rapidly, resulting in reduced colony survival. 22. Diet. Different species of Hymenoptera show a wide range of feeding habits. The most primitive forms are typically phytophagous, feeding on flowers, pollen, foliage, or stems. Stinging wasps are predators, and will provision their larvae with immobilized prey, while bees feed on nectar and pollen. A huge number of species are parasitoids as larvae. The adults inject the eggs into a host, which they begin to consume after hatching. For example, the eggs of the endangered Papilio homerus are parasitized at a rate of 77%, mainly by Hymenoptera species. 23. Some species are even hyperparasitoid, with the host itself being another parasitoid insect. Habits intermediate between those of the herbivorous and parasitoid forms are shown in some hymenopterans, which inhabit the galls or nests of other insects, stealing their food, and eventually killing and eating the occupant. 8. Classification. Symphyta, without a waist, the Sawfly arge pagana. Apocrita, with narrow waist, the wasp Vespula germanica. The Hymenoptera are divided into two groups, the Symphyta which have no waist, and the Apocrita which have a narrow waist. 4. Symphyta. The suborder Symphyta includes the sawflies, horntails, and parasitic wood wasps. The group may be paraphyletic, as it has been suggested that the family Oricidae may be the group from which the Apocrita arose. They have an unconstricted junction between the thorax and abdomen. The larvae are herbivorous, free-living, and eruciform, usually with 3 pairs of true legs, prolegs on every segment, unlike Lepidoptera and Acheli. The prolegs do not have crochet hooks at the ends unlike the larvae of the Lepidoptera. 4. The legs and prolegs tend to be reduced or absent in larvae that mine or bore plant tissue, as well as in larvae of Pamphyliidae. 24. Apocrita. The wasps, bees, and ants together make up the suborder, and clade Apocrita, characterized by a constriction between the first and second abdominal segments called a wasp waist, petiole also involving the fusion of the first abdominal segment to the thorax. Also, the larvae of all Apocrita lack legs, prolegs, or Acheli. The hindgut of the larva also remains closed during development, with feces being stored inside the body, with the exception of some bee larvae where the larval anus has reappeared through developmental reversion. In general, the anus only opens at the completion of larval growth. 4. Threats. Hymenoptera as a group are highly susceptible to habitat loss, which can lead to substantial decreases in species richness and have major ecological implications due to their pivotal role as plant pollinators. 25. Consumers are becoming more concerned about pesticide usage on ornamental plants and turfgrass in and around their homes and on the fruits and vegetables they eat. Not only are the negative health and environmental risks of pesticides of concern but also the impacts of neonicotinoids and other broad-spectrum pesticides on pollinators and other beneficial organisms. Growers and green industry professionals are searching for alternative pest management tactics to satisfy consumer demands and the desire for sustainability and operational flexibility. Many are considering biological control. The benefits of biological control include reduced reliance on pesticides, decreased potential for development of pesticide resistance, flexibility in usage of personal protective equipment, shorter or no restricted entry intervals, and reputational benefit of being a sustainable and responsible grower or professional. Biological control can also be used to manage pest populations that have developed pesticide resistance. This publication provides an introduction to biological control and explains how to integrate biological control into an integrated pest management (IPM) program. For the purposes of this publication, pests are defined as any undesirable insect, mite, plant, weed, or organism that causes disease, pathogen, or damage on ornamental plants, turf grasses, fruits, and vegetables. What is biological control? In nature, organism populations suffer frequent attacks and high mortality rates from predators, parasites, parasitoids, and diseases, collectively called natural enemies. Biological control tactics use natural enemies or agents, some practitioners call them beneficials, to manage pests. The ultimate goal of biological control is to suppress pest population and damage without pesticide or with reduced pesticide use. Natural enemies are utilized differently depending on the target pest, host, environmental condition, and pest life cycle. There are three general approaches to biological control. Classical biological control. Classical biological control refers to the practice of introducing one or a group of natural enemy species of foreign origin to control a pest that many times is also foreign in origin, called exotic, introduced, or invasive. Often, the natural enemies are found in the home range of the invasive pest. Some notable examples of classical biological control include the use of decapitating flies, several pseudactian species, against red imported fire ants, and a group of flea beetles, thrips, and stem borers used against alligator weed. Because of the long, rigorous, and costly process of finding, testing, quarantining, and rearing these natural enemies, classical biological control programs are typically conducted by scientists at governmental agencies or universities with public funding. Modern classical biological control programs mandate extensive testing of the natural enemy host ranges before introduction so that the selected natural enemies attack only 1. The intended target pest and do not cause harm to other non-target organisms. Once released into the environment, these selected natural enemies spread and manage the pest population with minimal assistance and intervention from the practitioners. The practical application of classical biological control by growers, professionals, and consumers on ornamental plants, turf grasses, fruits, and vegetables is minimal. Fortuitous or inventive biological control is a variant of classical biological control where natural enemies arrive from elsewhere by their own means and control the exotic pest population. These inventive natural enemies may have arrived with the pest or at a later time without introduction. There are also incidences where native natural enemies switch to using invasive pests as food or host on their own. Augmentative biological control. Augmentative biological control refers to the practice of releasing biological control agents, often mass-reared in insectaries, into an area where natural enemies are not present or present at a number too low to suppress a pest population. The goal of augmentative biological control is to increase the number or the effectiveness of natural enemies in an area to a level high enough to control the pest population. This type of biological control is most often practiced in greenhouses, nurseries, and some fruit and vegetable fields. The mass-produced biological control agents are purchased from the suppliers and released, applied en masse into the infested area to kill the pests. Biological control agents mass-produced by insectaries are often host-specific, i.e., they only attack one or two kinds of pests. Therefore, practitioners need to identify the pest species accurately so that the correct natural enemy species can be purchased for release. Depending on the pest and biological control agent species, as well as the environment and production practices, augmentative biological control can be achieved through inoculative releases or inundative releases. In inoculative releases, the biological control agents are released in small numbers to establish a population that provides long-term and sustained suppression of the pest population. In inundative releases, the biological control agents are released in large numbers to quickly overwhelm. In inoculative releases, the biological control agents are released in small numbers to establish a population that provides long-term and sustained suppression of the pest population. In inundative releases, the biological control agents are released in large numbers to quickly overwhelm the pest population without the expectation of propagating the biological control agent population or continuing the suppression of the pest population. Conservation biological control. Conservation biological control refers to a collection of methods and approaches in manipulating the habitat, plant diversity, production practice, and pest management practice to increase the population and effectiveness of natural enemies. An area with more complex and diverse plant and animal communities is known to have a greater diversity of natural enemies and a lower abundance of pests. Conservation biological control practitioners often start with manipulating the farmscape or landscape, such as growing insectary plants, i.e., plant species that can attract and retain natural enemies or provide natural enemies with food and shelter. Natural enemy diversity, abundance, and effectiveness increases plant diversity and resources provided increase. Other conservation biological control practices seek to minimize impacts of habitat manipulation or farming practices on natural enemies. For example, growers or landscape care professionals may use mulch to provide shelters for ground beetles, or reduce pesticide use, or use pesticides that have minimal impacts on the natural enemies, i.e., compatible pesticides. Biological control is a complex pest management strategy that requires a comprehensive understanding of the ecology and behavior of pests and natural enemies. As a result, biological control is often more difficult to design and put into action than simply spraying pesticides, chemical control. Biological control can sometimes be more expensive than conventional. 2. 3. 4. Chemical control. When designed and implemented correctly, however, the benefits of biological control in terms of environmental sustainability, efficacy, and cost-effectiveness can outweigh these shortcomings. Types of biological control agents. Natural enemies of insects and mites generally fall into 4 different types, or guilds, based on how they utilize their prey or hosts: predators, parasites, parasitoids, and pathogens. Predators are organisms that feed on the target pests and include insects such as lady beetles, green lacewings, rove beetles, hoverflies, and predatory mites. Table 1. Parasites and parasitoids are interchangeable terms for some practitioners, but there are significant differences between the two types. Typically, parasites are microorganisms that live, feed, and lay eggs on or in a host without killing it. Parasitoids do the same as parasites but eventually kill the host. Parasitoids are typically parasitic insects such as tachinid flies or parasitic wasps. Table 1. Pathogens include microorganisms, such as fungi, bacteria, nematodes, and viruses that cause diseases in pests. Pathogens that are used against insects and mites are referred to as entomopathogenic. Natural enemies of plant pathogens are generally microorganisms similar to their targets, i.e., fungi, viruses, and bacteria. These microorganisms interact with plant pathogens in four primary ways: competition, hyperparasitism, induced resistance, and production of antimicrobial compounds. Competition is relatively straightforward. A large number of beneficial microorganisms is applied to the environment, which takes up all the available living spaces or resources and denies occupancy by plant pathogens. Bacillus subtilis is a common example, where products containing this bacterium are applied to soil or soilless growing medium to outcompete root rot causing pathogens. Table 2. Hyperparasitism occurs when a beneficial microorganism parasitizes and eventually kills a plant pathogen. Some beneficial microorganisms can induce or cause plants to produce defensive chemical compounds to fend off pathogens, i.e., resistance. Hyperparasitism and induced resistance are very specific interactions among plants, beneficial microorganisms and pathogens, but are not widely utilized commercially. Production of antimicrobial metabolites that stop growth or kill the pathogens is the most common way biological control is used for disease management. The beneficial microorganisms, often bacteria, are mass-reared in fermentation vessels to produce specific antimicrobial compounds, which are later extracted and used as antimicrobial pesticides. The beneficial microorganisms can also be applied to the plants. When the beneficial microorganisms die, their cells release the antimicrobial compounds onto the leaf surface, thus killing the pathogens nearby or protecting the leaf from infection. Once again, some B. subtilis products are examples of using antimicrobial compounds. These products are sometimes mixed with copper-based fungicides to enhance the effect. While some biological control agents or beneficial microorganisms can feed or parasitize a large number of pest species, some are effective against only one or two species of pests. For example, the predatory mite Amblysia swerskii can feed on thrips, whiteflies, broad mites, and spider mites, whereas another predatory mite, Phytoceolus persimilis, only feeds on spider mites. Table 1. Differences also occur within the same biological control agent species. For example, Bacillus thuringiensis subsp. Kerstaki is effective only against caterpillars, but Bacillus thuringiensis subsp. Gallerii is effective only against white grubs. Table 1. Because the relationships between biological control agents and pests can be quite specific, accurate identification of the pest organism or disease, often to the species level, is crucial to selecting the correct biological control agent species and the success of the biological control program. 5. When biological control agents are introduced or released into an environment, their application methods differ among species. Larger agents, sometimes referred to as macrobiological control agents, such as predatory mites and parasitoids, are applied in loose carrier, usually brand, sachets, or cards adhered with eggs or pupae. Microbial biological control agents, sometimes referred to as microbiological control agents, are often diluted in water and sprayed onto the plants or drenched onto medium or soil. Biological control is part of an IPM program. No pest management tactic acts independently in an IPM program. The same principle applies to biological control tactics. For example, pest species must be accurately identified to select the correct biological control agents or microbial products. Also, biological control is most effective against a small pest population or when pesticides that can negatively impact the survival and functions of the biological control agents are removed from the program. Biological control must be an integral part of an IPM program, where biological control tactics are adopted in consideration of other tactics within the program and vice versa. An IPM program starts with scouting. In an IPM program that incorporates biological control, both the pest and natural enemy populations must be sampled, and their densities or abundance determined. Biological control performs more effectively when biological control tactics are applied when the pest population is still small. The release of biological control agents does not signal the end of scouting but a need for continuation so that other pest management tactics can be applied when the need arises. Successful biological control will suppress the pest population to a level that would not cause damage, and therefore, pesticide use can be delayed or avoided completely. However, when the pest population increases above the economic or aesthetic threshold, or the biological control agent population or effectiveness decreases, pesticides may have to be applied to suppress the pest population and damage and allow biological control to catch up. Cultural and production practices that growers employ may also impact biological control. There are often more natural enemies and greater biological control effectiveness when the fields or landscapes contain more abundant and diverse vegetation. Growers and landscape designers should strive to practice environmental engineering or production practices that incorporate many plant species, particularly those that can provide resources to the biological control agents. Intercropping and cover cropping with insectary plants help attract and retain biological control agents and serve to reduce weed management needs and costs, improve soil health, and control soil erosion. Maintaining vegetation and mulch debris allows biological control agents to find refuge and resources and maintain a consistent ever-present population that can disperse as pest management needs arise. Which pesticides are used, and how and when, has a significant impact on the success of biological control. Biological control agents are as susceptible as, and in some cases, more sensitive to pesticides than their target pests or diseases. Broad-spectrum pesticides can kill or affect a wide variety of biological control agents. For example, organophosphate and pyrethroid insecticides are very toxic to many biological control agents and pollinators, and fungicides that target multiple sites, such as copper and mancozeb, can reduce the effectiveness of beneficial microorganisms. These pesticides should not be used in conjunction with biological control. Instead, pesticides that have a reduced risk or lower toxicity to the biological control agents, commonly referred to as the compatible pesticides, should be used. Additionally, some pesticides may also destroy habitats of the biological control agents and should not be used. For example, the use of broad-spectrum herbicides and other indiscriminate weed management practices must be eliminated or carefully considered if companion plants are used. How 6. Pesticides are applied also impacts the success of biological control. Application methods, such as drench, and timing, depending on the biological control agents, that avoid direct contact or leave behind large amounts of residue on the plant's surface should be used. Biological control is a knowledge-intensive strategy of pest management. Successful implementation of a biological control program requires a thorough understanding of the pests, the natural enemies, their environment including other pest management practices, and the interactions of all factors. Success is often achieved after many considerations, modifications of current production, and pest management practices, as well as trial and error. Despite being challenging to adopt, biological control and IPM, in general, create benefits that contribute to building a sustainable environment and increasing profitability by reducing management inputs. Table 1. Commercially available biological control agents for major arthropod pests. Type species commonly known as target insects and mites. Predatory mite Amblydromalis limonicus. Limonicus mite thrips, whitefly. Predatory mite Amblysius andersoni mite two-spotted spider mite, russet mite, rust mite. Broad mite. Predatory mite Amblysius degenerans mite thrips, broad mite. Two-spotted spider mite Predatory mite Amblysius swirsky mite thrips, whitefly, and broad mite. Predatory mite Galindromus occidentalis. Galindromas mite two-spotted spider mite, aerophede mite, russet mite. Predatory mite Mesoceolus longipes, formerly Phytoceolus. Longipes. Longipes mite two-spotted spider mite. Predatory mite Neoceolus californicus, formerly Amblysius. Californicus. Californicus mite two-spotted spider mite, broad mite, cyclamen mite. Predatory mite Neoceolus cucumaris, formerly Amblysius. Cucumaris. Cucumaris mite thrips, fungus gnat, two-spotted spider mite. Tarsonemid mite. Predatory mite Neoceolus follicis, formerly Amblysius. Follicis. Follicis mite two-spotted spider mite. European red mite, citrus. Red mite. Predatory mite Phytoceolus persimilis mite two-spotted spider mite. Predatory mite Stratiolilaps simitus. Formerly Hypospis. Miles. Hypospis mite fungus gnat larva, thrips pupa. 7. Biological control strategies in integrated pest management, IPM programs. When biological control agents are introduced or released into an environment, their application methods differ among species. Larger agents, sometimes referred to as macrobiological control agents, such as predatory mites and parasitoids, are applied in loose carrier, usually brand sachets, or cards adhered with eggs or pupae. Microbial biological control agents, sometimes referred to as microbiological control agents, are often diluted in water and sprayed onto the plants or drenched onto medium or soil. Biological control is part of an IPM program. No pest management tactic acts independently in an IPM program. The same principle applies to biological control tactics. For example, pest species must be accurately identified to select the correct biological control agents or microbial products. Also, biological control is most effective against a small pest population or when pesticides that can negatively impact the survival and functions of the biological control agents are removed from the program. Biological control must be an integral part of an IPM program, where biological control tactics are adopted in consideration of other tactics within the program and vice versa. An IPM program starts with scouting. In an IPM program that incorporates biological control, both the pest and natural enemy populations must be sampled, and their densities or abundance determined. Biological control performs more effectively when biological control tactics are applied when the pest population is still small. The release of biological control agents does not signal the end of scouting but a need for continuation so that other pest management tactics can be applied when the need arises. Successful biological control will suppress the pest population to a level that would not cause damage, and therefore, pesticide use can be delayed or avoided completely. However, when the pest population increases above the economic or aesthetic threshold, or the biological control agent population or effectiveness decreases, pesticides may have to be applied to suppress the pest population and damage and allow biological control to catch up. Cultural and production practices that growers employ may also impact biological control. There are often more natural enemies and greater biological control effectiveness when the fields or landscapes contain more abundant and diverse vegetation. Growers and landscape designers should strive to practice environmental engineering or production practices that incorporate many plant species, particularly those that can provide resources to the biological control agents. Intercropping and cover cropping with insectary plants help attract and retain biological control agents and serve to reduce weed management needs and costs, improve soil health, and control soil erosion. Maintaining vegetation and mulch debris allows biological control agents to find refuge and resources and maintain a consistent ever-present population that can disperse as pest management needs arise. Which pesticides are used, and how and when, has a significant impact on the success of biological control. Biological control agents are as susceptible as, and in some cases, more sensitive to pesticides than their target pests or diseases. Broad-spectrum pesticides can kill or affect a wide variety of biological control agents. For example, organophosphate and pyrethroid insecticides are very toxic to many biological control agents and pollinators, and fungicides that target multiple sites, such as copper and mancozeb, can reduce the effectiveness of beneficial microorganisms.